Okay, today we shall see DNA replication in eukaryotes. Last time in my video, we discussed how replication processes then in prokaryotic cell. You know, two types of cells, prokaryotic and eukaryotic. When you see prokaryotic, like algae, fungi, plant and animal cells, how replication will be carried out in this type of organisms, we will see. So, what are the mechanisms? What are the role of different enzymes? You know, eukaryotic cell is organisms that possess defined nucleus. In the cytoplasm, there is nuclear membrane bounded nucleus, in addition to another organelles like mitochondria, reticulum endoplasmic, Golgi apparatus, lysosome, vacuoles. No such thing in prokaryotic cell. So the special feature here in eukaryotic is the chromosome is found in nucleus. This chromosome is a linear. It is a linear type of chromosome. It is not circular. It is multiple. Unlike prokaryotic, you know, in prokaryotic it is a circular chromosome, but here it is a multiple linear chromosome. Again, there is a centromere on the chromosome. There is a telomeres at the end of the strand. You know, on the chromosome, DNA and histone proteins are found. Just DNA is wrapped around histone protein. Anyway, here the replication process it is very complex process. It involves so many enzymes. Look at here. You know, on the chromosome, this is a chromosome. Here there is histone protein and the DNA. This one is a histone. Here a DNA. This tree-like structure is a DNA which is wrapped around the histone protein. You know, replication process is determined by accessibility of DNA to the enzymes. If they are tightly wrapped around histone protein, it means that it cannot be accessible to proteins to start replication. So replication is depend upon accessibility of DNA. You know, it is very super cold here. DNA and histone protein tied together, it form nucleosome. This is helix structure. So anyway, the process is very complex here in eukaryotic. What are the mechanisms? The basic process is the same. There is initiator protein that bind at the origin of replication. But here in eukaryotic, there is multiple origin of replication here. For example, in yeast, 400, in human being, more than 10,000. Imagine how much is very complex. In prokaryotic, there is only single origin of replication. The same is true here. There is helicase enzyme. Helicase enzyme unwinds and separates DNA helix into single-stranded DNA. So replication fork will be formed. There are so many enzymes. We'll see anyway. Here, topoisomerase, its responsibility is it relaxes supercoiling of DNA. DNA is super coiled. It has to be relaxed. It has to be relaxed. If not, DNA helices cannot unwind, cannot break the bond. So this is the role of topoisomerase enzyme. It relaxes the strand. Then helicase enzyme start unwinding the strand. It break the bond. It cut the bond. After that, SSB protein, single-stranded binding protein, keep the strand apart. It attached to each strand, then it remains separate. It remains separate. That is the role of SSB protein. Look at here. There is multiple origin of replication in eukaryotic, mind you. These are origin of replication. These also origin of replication. These are so many origin of replications are found here. So imagine how much the process is very complex, very complex. There is multiple origin of replications. There is so many enzymes. Okay? Actually, in case of prokaryotic, all things goes together. Goes together. But here, sometimes after replication, after separation is complete, then synthesis will begin sometimes. So anyway, new strand is formed from this. Mind you, here there is multiple origin of replication. So after that, after it is initiated, after the strand began separating, enzyme DNA polymerase start adding new nucleotide upon the existing started strand. For that matter, primers are very important. Mind you, replication requires 4 things: strand of DNA polymerase, substrates, that is nitrogenous bases, and the primers. Primers are short strand of RNA that is important to start replication, because primers do have hydroxyl group. It provides OH. So at that region, new nucleotide will be attached, phosphate will be attached there. So that primers are very important to start replication. So after replication began, the enzyme flap endonuclease 1 and RNase H remove a primers at the start of leading strand and at each start of lagging strand, which is Okazaki fragment. A lagging strand, it is some kind we call Okazaki fragment. There are many strands. For each strand, different primers are very important. So all these primers, both from leading and lagging strand, will be removed by flap endonuclease 1 and RNase H. Then after, DNA polymerase delta aid in elongation of lagging strand. After primers are removed. Mind you, DNA polymerase alpha for the initiation provide primers. Now primers are removed. Then DNA polymerase delta elongate lagging strand. In addition to this, it do have exonuclease activity. It means proofreading. There is some error. Actually, it is a rare case. DNA replication is very, very accurate. It's very, very accurate. It is very, very accurate. The process is carried out with very high fidelity. It means there is a proofreading. There is a correction of different errors if it probably happened. Actually, this is a rare case. So here, this enzyme, DNA polymerase delta, in addition to lagging strand elongation, it solved error, mismatched base pairs at the primer's terminus. DNA polymerase epsilon, it aid leading strand elongation. It add new nucleotide on growing leading strand. That is the role of DNA polymerase epsilon. In addition to that, it do have regulatory role. Actually, it is highly related to polymerase delta. DNA polymerase beta, it helps in repairing mechanism. Again, base excision repair. Base excision repair. If there is error, if there is mismatch, if there is some deletion. Actually, this is a rare case. Anyway, this enzyme help in repairing mechanism. It repair. It repair. So now, mind you, during elongation, there is adding of new nucleotide on existing strand. In such a way, it become elongated. It become very huge and a complex molecule. So enzymes, different enzymes, polymerase enzymes help in adding new strand. Leading strand is synthesized continuously from 5 to 3 direction. Lagging strand is synthesized discontinuously in direction away from the replication fork, in Okazaki fragment. Actually, the number of Okazaki fragment here in eukaryotic is 102 to 200 base pair. Each do have its own primer, I said already, because the DNA polymerase can only synthesize in the 5 to 3 direction. So it constantly encounters the previously synthesized new strand here. Look at here. This is a strand. Now, here around this, there is topoisomerase enzyme. That topoisomerase enzyme, it relaxes this strand. You know this strand is, mind you, it is twisted, it is rapid, it is supercoiled. DNA is wrapped around histone protein. If it is not relaxed, replication never begins. So a topoisomerase enzyme, it relaxes this strand. Then helicase enzyme, it separates this strand, began separating the strand. After helicase separates this strand, SSB protein bind upon each strand. Look at it. This is SSB protein. This is SSB protein. Single strand binding protein. So it keeps separate. Because it is attached on each of strand, so that strand remains separate. After that, leading strand is synthesized continuously from 5 to 3 direction. Mind you, here, this is from 5 to 3 direction. This should be from 5 to 3 direction. It is antiparallel. So leading strand is synthesized continuously. Just DNA polymerase epsilon simply add nucleotide. This DNA polymerase epsilon, it aids in elongation of leading strand. Simply it adds new nucleotide because the direction of synthesis is toward replication fork. You know, at the 3 end, there is free hydroxyl group. Simply new phosphate will come and attach. New nucleotide will come and attach. In such a way, elongation is carried out. Then, here, DNA polymerase delta aid in lagging strand elongation. Mind you here, since it is away from the replication fork, it is in the form of Okazaki fragment. This is a replication fork. If this is from 5 to 3 direction, this should be from 5 to 3 direction. Mind you, the strand should be synthesized, the new nucleotide should be added at the 3 end. 3 end is found here. So, DNA polymerase delta come and synthesize this. After it finish, it come back and start synthesize here. After it finish, it come back and synthesize here. After it finish, it come back and synthesize here. So, Okazaki fragment will be formed. That Okazaki fragment is filled by DNA ligase enzyme. Mind you, for each strand, different primers are very important. Here, around this, there is a primer. This one is a primer. these are primer, and so on, and so on. So the question is, how these primers should be removed by enzymes? How it will be filled? DNA polymerase simply fill this gap, the place where primers are removed. DNA polymerase simply fill the gap. But at the 5' end, primers will be removed. That gap remained. DNA polymerase cannot fill. Not only this, around this, there is gap here. Because at the 5' end, there is no OH group. So nucleotide cannot be added here because there is no OH. So this gap remain. This gap remain. This gap remain. Again, this gap remain. Remain unpaired. Remain unpaired. How it will be formed? How it will be formed? This is the question actually. You will see anyway. Mind you, here primers are removed at the 5' end. The primers between fragments are simply synthesized by DNA polymerase. But the primers at the 5' end remains unsolved. Primers at the 5' end of leading strand and also remained. These primers may cause loss of terminal sequence. How these gaps are solved? How these gaps are filled? Let us see. After new strands are synthesized, there is what you call a termination. Mind you, the cell know how to start and how to terminate. In a termination site, there is many GC content. It is rich in GC rich region. So around that, replication may be terminated. Actually, two things are very important. The first is end replication problem. The other is protection of DNA telomer. At the 5' end, there is telomere. At the end of strand, there is a telomere. How to protect it? So let us see. In eukaryotes, removal of terminal primer at the end of lagging strand leaves a gap that cannot be filled if left unmixed. This gap leads to loss of terminal sequences. Loss of terminal sequence, especially at the 5' end. This is known as end replication problem. End replication problem. For that matter, there is an enzyme telomerase, telomere terminal transferase. This enzyme is ribonucleoprotein. It do have protein and RNA template. Telomere serve to protect genetic information contained at the end of eukaryotic chromosomes. It extends 3 ends of the parental chromosome beyond 5 ends of the outer strand. The extended strand is not used for protein synthesis. It is a junk element. Here, telomeric repeat binding factor bind double-stranded telomeric DNA and have a role in telomere stabilization and telomere length regulation. So there is protruding 3' can invade duplex DNA and form laurent-like structure, which is a T-loop. So it helps in protection. Look at here example. This is a strand. This is a parental strand. These two are this one and this one is a parental strand. This is a 5' end. This is a 3' end. This is the 3' end. At this 3' end, there is a telomere. There is a telomere. This is a telomer. TGG, GG, TG. This is a telomer. This sequence is a telomer. But the question is, the gap on fragment, the between fragments can be synthesized, can be filled by DNA polymerase. What about the gap left at the 5' of lagging strand and at the leading strand? This is the question. How it will be filled? If not, it causes a terminal sequence. It causes a terminal sequence. For this matter, at the 3 end of leading strand, there is a sequence of telomer, TGG. For this matter, an enzyme telomerase solve this issue. You know this telomerase is, it is protein and RNA template. Protein and RNA template. RNA template, telomer do have complementary sequence to that of telomer. This is a T, this is a, GGGG, CCC, TT, AA, G, C. So it provides this complementary sequence here. This is existing telomer. Then here it provides A instead of T. It provides C. Okay. Then this sequence. After that, upon this sequence, this will be synthesized. This will be provided. G, 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 T, T, G. Okay? Yeah. T, T, G. Again, G, G, T, T, T. So it do have a complementary sequence. It bring and attach at this region. Then this telomere elongate. Again, it bring and attach at this region. Again, it bring A, C, 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 A, C, then this also elongate. In such a way, it form very, very huge strand at the end. That strand is not used for coding, rather it is important for the protection. It is a junk element. It is a junk element. It is a junk element, not used for coding. Rather, it is important for the protection. It is important for the protection. So this telomere enzyme solve end replication problem of eukaryotic cell type. It provides complementary sequence. So the loss of terminal sequence problem can be solved in this way also. In this strand also the same is true. Just here is a telomere around the 3 end. Then the complementary sequence will come. Then in such a way it will be elongated. So end replication problem can be solved. As a summary, you know, prokaryotes occur. In prokaryotes, replication occur inside cytoplasm, but here in nucleus. You know, in prokaryotic, all things goes together. Replication, once replication begin, transcription start. Once transcription start, protein synthesis start. But in eukaryotic, is a different. Actually, I will provide another video of transcription anyway. There is only single origin of replication in prokaryote. These do have many origins of replication. Each origin of replication is formed of about 115 nucleotide. This one, 102, 200 or more. Initiation is carried out protein DNA A capital and DNA B capital. So initiation is aided by this. Here initiation is carried out by ORC origin recognition complex. This origin recognition complex binds to DNA at replication origin sites, serve as scaffold for the assembly of other key initiation factors. So this is how replication is carried out in eukaryotic. Mind you, in eukaryotic, the process is very complex. It's not like that of prokaryotic, like a bacteria. Many more enzymes are. The structure itself is very complex. So this is a difference between prokaryotic and eukaryotic. Thank you for watching this video. Share, subscribe, and comment on this.